These are the lecture notes for Chapter 12 on the cell cycle, and this is primarily going to be on the process of mitosis. So basically, when mitosis occurs, or when a cell divides, a eukaryotic cell, and I'm just talking about eukaryotic cells here that have a nucleus and organelles. So basically, any kind of cell from the kingdom Protista, plants, animals, and fungi. Okay, so the cell that's about to divide, we're going to call that the parent cell. And before it ever divides, it's going to have, if this were a human, instead of one chromosome here, we would see 46, okay? 46 single-stranded chromosomes. So right before the cell divides, it is going to duplicate each chromosome so that now it's got like an identical copy. We call these sister chromatids. So both of these are literally identical DNA codes. So you're going to have 46 duplicated chromosomes right before the cell begins to divide. Okay, that happens in interphase. And then the actual cell division part is going to be divided into two different stages. Mitosis is the division of the nucleus and separation of the chromosomes. So each daughter cell gets a copy of all the DNA that was in the parent cell. Because mitosis is cloning. This isn't, this is going to be taking one cell and splitting it in half, making two new cells, and each new cell is genetically identical to that parent cell. So if the parent cell had 46 chromosomes, then the daughter cells are going to have 46 chromosomes. So the cell division part is mitosis, and that is when the sister chromatid copies, chromosome copies called sister chromatids, they are separated and they move to opposite ends of the cell. And then when the cell splits in half by cytokinesis, each daughter cell is genetically identical to the parent. That's mitosis. Now, in Chapter 13, we're going to learn about meiosis, which is when the daughter cells only have half of the DNA of the parent. But this is mitosis, and mitosis is cloning. This is a video of a sea urchin embryo. And that's on slide 4. So it's just going to show you cell division in the sea urchin. And it says the continuity of life is based on the reproduction of cells or cell division. And the purpose of cell division and mitosis is if it's a single-celled organism, whether that is a bacterium or an archaea, which are prokaryotes, they're not going to undergo mitosis, but they are able to divide by a process called binary fission. But and the other single-celled organisms would be the protozoans, which are still single-celled, but they're eukaryotic cells. So they do divide by mitosis. But the reason single-celled organisms divide is to make new organisms. So single-celled organisms give rise to new organisms through cell division. But multicellular eukaryotes, which would include from the kingdom Protista, that would be the algae. And then the kingdom Plantae, the kingdom Animalia, and most of the members of the fungi. Okay? So when multicellular eukaryotes, which start out as a single cell called a zygote, when that zygote begins to divide, We call that embryonic development. So when multicellular eukaryotes undergo embryonic development, and they start going from a single cell into, sometimes like with humans, trillions of cells. That is one of the roles of cell division. And the other thing is renewal and repair. Renewal, repair, and then growth. The growth kind of goes along with embryonic development all the way through like adulthood. After the baby is born and becomes an adult, all of that development, growth and development. But then in addition to that, renewal and repair. So if you are injured, if you lose tissue, the cells in some tissues can divide and replace the ones that were lost. And of course, there are tissues that are constantly shedding cells and replacing them, like the skin and bones. Bones actually do that too. They just don't do it as often as the skin. All right. All of the DNA in a cell constitutes the cell's genome. So all the DNA in a human cell is going to be found in 46 chromosomes. So all 46 chromosomes together, including the genes that are within the chromosomes, that would be the genome, okay? DNA molecules in a cell are packaged into chromosomes, and a chromosome can carry up to a few thousand genes. You can see a picture here of a eukaryotic cell, and you can see inside, and this, I can look at this and tell you what stage of mitosis it's in. Also, it's in prophase. But you see, all of these chromosomes look like spaghetti strands. So we're going to talk about two different forms of DNA within the cell. There's chromatin, which is all spread out. It's not this. It's all spread out. You can't make out individual strands. You can't see individual strands. And then there's chromosomes, and chromosomes form from the chromatin during mitosis. All right, so eukaryotic chromosomes consist of chromatin, and chromatin is actually not just DNA. It's a complex of DNA wrapped around protein. And then when the cell is dividing, the chromatin condenses or coils into rod-shaped structures called chromosomes. Somatic cells are body cells. Somatic cells are all the cells in the body that are not egg and sperm, okay? Somatic cells have two sets of chromosomes. The gametes, which would be the egg and the sperm, have half as many chromosomes as somatic cells. So human somatic cells have 46 chromosomes or 23 pairs, and human gametes have just 23, half as many chromosomes as somatic cells. All right, before the cell divides, the chromosomes duplicate. So each duplicated chromosome has two sister chromatids, which are identical copies. And this will happen during interphase, before mitosis. And the sister chromatids are attached by cohesins. And the place where they're attached kind of looks like a waist, and we call it the centromere. And you can see this is from a scanning electron microscope. So it's an image from a scanning electron microscope, which is much better for looking at the surface of structures. But this is a duplicated chromosome. You can see the centromere, which is the area where they're connected, and you can see that each strand is a sister chromatid. but the whole thing is one chromosome. You don't count it as two. You count it as one chromosome. In fact, let's go back to this picture because sometimes this is hard for students to get. In the parent cell, what you're looking at is one single chromosome. In the cell below the parent cell, you're still looking at a single chromosome. The only difference is in the parent cell, it's a chromosome that hasn't replicated, and in the cell below it, it's a duplicated chromosome. But either way, they count as a single chromosome, because even when it duplicates, it's not like extra genetic material. It's identical copies of the same chromosome. Not just identical traits are found in the chromosomes, but actually identical versions of the traits. So if you had I'm sorry, I should have just stayed there. If you had the gene for eye color on this sister chromosome on the left, and let's say it was for, I don't know, green eyes, then the one on the right would also have a gene for green eyes. It wouldn't have a gene for blue or brown or any other color. It would have the gene for green eyes identical. So they're just, literally sister chromatids are identical copies. Okay. So this is just showing you again about the duplication, and then you've got an animation on the entire process of mitosis, and this is slide number 16. Now, cell division in eukaryotic cells consists of mitosis and cytokinesis. Mitosis technically is the division of the genetic material or the chromosomes in the nucleus, whereas cytokinesis is when the cell actually splits into two cells, or the division of the cytoplasm. So mitosis is division of the nucleus, and cytokinesis is the division of the cytoplasm. And actually cytokinesis occurs at the end of mitosis, but it's like as mitosis is ending, cytokinesis is occurring. So the last stage of mitosis and cytokinesis happen together. All right, now the production of gametes, the production of egg and sperm occurs by a process called meiosis, and some people say meiosis. I've just always said meiosis, and we'll talk about that in chapter 13. All right, the cell cycle, not cell division, but the cell cycle. That means the cell cycle is all the events that occur from the time a cell is produced until it divides again. So the cell cycle is going to consist of the M phase or mitotic phase, and that will be mitosis and cytokinesis. So that's the cell division part. And then interphase, which is when the cell is not actively dividing, but it is growing, and it is duplicating the chromosomes and preparing for cell division. Okay, so first we'll talk about interphase. If you looked at the cell cycle and you counted the amount of time that a cell spends in interphase compared to the amount of time it spends in mitosis, interphase is going to be about 90% of the cell cycle. So almost all the time, cells are in interphase. There's 3 phases of interphase. There's G1, S for synthesis, and G2. I know it says G1 is first gap and G2 is second gap, but I always think of the G as growth. But whatever works. S stands for synthesis, and that's the phase where the DNA is duplicated. And then here's a circle or a pie graph that shows you pretty much the amount of time the cell spends in all the phases. So interphase, as you can see, takes up about 90% of the life of a cell. And then the cell division part, the mitotic or M phase, consists of mitosis and cytokinesis. And then moving back to G1, S, and G2. In the G1 phase, you have metabolic activity like cellular respiration and that kind of thing, and growth. So what happens specifically in the inside of the cell is that not only does the parent cell have to copy its DNA, it also has to make a copy of every organelle so that the daughter cell will have a copy of every organelle that the parent cell has. And that is what happens in G1. In G1, the cell doubles its organelles. So if it has one mitochondria, it doubles that mitochondria. Then the S phase is when DNA replicates, so it's DNA synthesis. It's when the single chromosome becomes two sister chromatids. And the G2 phase, a lot of different things happen, but the cell is preparing to divide in G2. And one of the things it does is it starts to produce those microtubules called spindle fibers that are going to attach to the sister chromatids and pull them apart. All right, your 5 stages of mitosis are prophase, prometaphase, metaphase, anaphase, and telophase. And some people say telophase. I don't. I say telophase. So mitosis has 5 stages. And these are hard. These pictures are hard to, I like the illustrations a little bit better. All right, so in interphase, if you looked at a cell under the microscope, you would see a solid color, a solid nucleus. You wouldn't even see spaces. You wouldn't be able to see the chromatin. It would really be like just solid blue, because the DNA is in the form called chromatin, and it's all spread out. So it would just look like a solid nucleus inside the cell. In prophase, you're going to have the nuclear envelope starting to disappear. The chromosomes, or the chromatin, condenses into chromosomes. So you can see the spaghetti strands. You can see the chromosomes. And you have these structures called centrioles that are going to start moving to opposite ends of the cell. And the spindle fibers, the microtubules we call spindle fibers, they also begin to form, and they're kind of extended between the 2 centrioles. Now, in prometaphase, only one thing happens. In prometaphase, the spindle microtubules attach to the kinetochore, which is a little structure right there at the centromere of each chromosome, each duplicated chromosome. So it's like just a little structure that the spindle microtubules can attach to. So the spindle fibers attach to the chromosomes in prometaphase. The chromosomes line up in the middle of the cell in metaphase. The sister chromatids are pulled apart or separated in anaphase. And the sister chromatids have moved to opposite poles by telophase. And the nuclear envelope has started to reform around those sets of chromosomes. And then, in addition to that, cytokinesis occurs, and the cell splits into 2 cells. Another animation on mitosis. This is slide 28. And then another one where you can watch mitosis happening in time lapse, like under a light microscope. So that is, and this would be a light microscope, and you know that because you can't see living cells with electron microscopes. So that is slide 29. A closer look at the mitotic spindle, and I'll cover this and then I'll end, and start a new recording. But anyway, the spindle is made of microtubules. So you'll hear me say microtubules, spindle fibers, or spindle microtubules. So it's a structure made of microtubules that controls chromosome movement during mitosis. And the centrosome is a microtubule organizing center, and it contains centrioles, at least in animal cells. Plant cells actually don't have centrosomes or centrioles. But the centrosome contains the microtubule organizing center, and it's going to produce the microtubules for mitosis. Now, each sister chromatid has a kinetochore. It's a protein complex that is located on the outside edge of the centromere, which is, the centromere is like the waist of the duplicated chromosome. It's the area where the sister chromatids are attached. So you have this protein complex called a kinetochore on the outside of each of those centromeres. And that is where the microtubules attach to the chromosomes. You see the little black structure? So the area where the sister chromatids are attached is called the centromere, which I don't see here. I don't see it labeled. But the little black structures on the outside of that are called kinetochores. And then the last thing I'll say is the way that the microtubules pull the sister chromatids apart in anaphase is that they depolymerize. In other words, they get shorter. Microtubules depolymerize, or they get shorter, and that causes the sister chromatids to separate and move to opposite ends of the cell. All right, so we'll stop here and we'll start on slide number 40 in the next recording.